So, good evening. This is Bio 212, Chapter 25, and we are working on Part C now of cellular respiration and metabolism. And what we're doing is we're about to move into what other outcomes of glucose, amino acids, and triglycerides could you have in normal metabolic pathways. But before we do that, I want to go over the summary of cellular respiration to remind people that glucose inside the bloodstream can be taken up inside of a cell, catabolized into pyruvic acid if oxygen is in sufficient concentration. That pyruvic acid inside the matrix of the mitochondria is converted into acetyl coenzyme A, and it's that acetyl coenzyme A that can now move into the Krebs cycle. The products of that glycolysis and the Krebs cycle will be shunted to the electron transport chain, where the electron transport chain utilizes the energy to break apart oxygen molecules. That's why you inhale oxygen, okay, to allow us to make ATP and metabolic water. Here are the total outputs for each of the individual steps that are listed for you over here. What we want to do, though, is we want to change our question a little bit. And that is, well, we don't always know when we're going to have glucose available to us. So what mechanism does Mother Nature have to either store glucose when it gets outside of its normal physiologic range, meaning we have too much of it, or break it down when we have too little of it? Okay? So that's what we're going to do here. And I'm going to draw for you this little squiggly line. And if you've taken a class with me before, you know what this means. And I know it's not perfect, but it'll work, right? And remember, you have a concentration of glucose inside your bloodstream all the time. Okay, remember your beta cells inside of your pancreas are measuring that. And if we have too much of it, it's a ripe opportunity to make hay, meaning store it. Okay? Glucose is anabolized into the polysaccharide, which is glycogen. That's all there is to it, right? And what that means is that polysaccharide is the only stored carbohydrate inside humans. We don't store cellulose. We definitely don't store starches. Okay? And the reason is its insulin is going to stimulate hepatocytes and skeletal muscle cells to stimulate glycogen. Here's the difference. Hepatocytes don't need insulin to get glucose into them. Skeletal muscle cells do. If insulin is in the bloodstream at high enough concentration, what's happening is the hepatocytes are being now told to take that glycogen and store it, I should say glucose, and store it as glycogen. Okay, so it's an incredibly important difference. Just one more time to make sure you understand it. Hepatocytes and your central nervous system. Okay, I'm going to put that there just to make sure you understand that. And CNS do not require insulin to bring glucose into their cells. Skeletal muscle cells do. However, if there's insulin inside the bloodstream at high enough concentration, hepatocytes and skeletal muscle cells will now take that glucose and convert it into glycogen. When there's insufficient glucose inside the bloodstream, meaning we're down here now, right? A signal will now cause the glycogen stored inside hepatocytes to be broken down into glucose molecules. And notice here the difference is it's being released into the blood. Skeletal muscle cells, when they break it down, they're using it for the metabolism of the muscle cell. So chances are not a whole lot of it's going to get into the bloodstream unless you've run really low. Okay. So how does this happen? Well, let's get rid of all this stuff first. Let's move to this slide here. And notice there's a color key here, and this color key looks awfully like that previous set of figures we looked at. Here's our purple glucose. Remember, if I drew the six-sided sugar, it's now broken into nothing here. Notice it's not converted into a 2,3 carbon structure. Glucose is brought inside the cell. Remember, we phosphorylate it to make sure it doesn't leave. It's then converted into glycogen. by making glucose 1-phosphate, which is converted into uridine diphosphate glucose. Don't worry about this part here. Just remember it's phosphorylated, and then it's converted into glycogen. What is important to worry about here, though, right? What is very important to worry about here is, well, if we need to store glucose, right, what are we going to do? We're going to use insulin to actually convert glucose into glycogen. Then, if the levels get too low, then we're going to use glucagon and epinephrine to cause the phosphorylase to convert glycogen back into glucose 1-phosphate, where you're going to dephosphorylate that glucose so it can now exit the liver to move into the bloodstream. So in red here, we see we use insulin. In blue here, you see we use glucagon and epinephrine. Okay. One more time, just to make sure you understand what you're looking at here. Stored glycogen inside of a cell got there because in the presence of insulin, excess glucose is converted into glycogen. In the absence of insulin and excess glucose inside the bloodstream, we use glucagon and epinephrine to convert that glycogen back into glucose, which can then leave the cell. Okay? And notice the cell here is the hepatocyte. Let's get rid of these cartoons. Let's move on. So what's going to actually happen? Well, I just showed you what happens with glucose, right? And you can actually see that glucose is here. Now notice glucose is inside the circulatory system. The purple arrows here mean that if we have a different formulation of, say, glucagon and cortisol, we can utilize lactic acid, specific subsets of amino acids, and glycerol to make glucose. They can be entered into a piece of the glycolytic pathway such that we end up releasing glucose into the bloodstream. So let's just make sure we understand this. First of all, it's called gluconeogenesis. So let's bold underline that for you. Okay. Number one. Number two, where glycerol, part of triglycerides, lactic acid, and certain amino acids are converted by the liver. So these are the hepatocytes. So the hepatocytes are responding to cortisol and glucagon so that you actually form non-carbohydrate sources of glucose. Not a carbohydrate, not a carbohydrate, not a carbohydrate. And in the presence of cortisol and glucagon, you can make a carbohydrate. Okay? There are some of you that would argue with me that glycerol could be a carbohydrate, but remember, glycerol means something else. That OL means something. So, how does Mother Nature actually do this? Well, let's think about lipid metabolism, right? We store non-soluble structures, meaning cholesterol, triglycerides, inside of this thing, okay? This thing could be a chylomicron. It could be a lipoprotein encoded vesicle that's holding all of these. The point is that they are transport vesicles. Why do we care about these? Well, take all of this with a grain of salt, because when this issue of the textbook was written, which was only a year and a half ago, all the reports of the concentrations of low density, very low density, and high density lipoprotein characterizations were being reformulated. But very low density lipoproteins are formed inside the hepatocytes, and they transport lipids to adipocytes. Low density lipoproteins, we used to call these things bad cholesterol, okay? They can deposit cholesterol in fatty plaques. It's completely true. Significance, though, unclear. And high-density lipoproteins, good cholesterol, remove excess cholesterol from body cells and blood, deliver it to the liver for elimination. Elimination can also be synthesis. Okay, so take all of this with a grain of salt. I think there's a lot more work that has to be done here before we actually understand that lipids are going to be used to make steroids. We need some steroids, okay? Meaning good cholesterol, bad cholesterol. The biochemistry still has to be worked out. Lipid metabolism, there are two sources of cholesterol inside the body. They're present inside your food, and like I said a moment ago, they can be synthesized inside the hepatocytes. Total blood cholesterol is going to increase as a result of lack of exercise, bad diet, etc., etc. Lipids can be oxidized. Here's that big fancy word again. I don't mean it's fancy anymore. Bold underline, okay? We can burn them, essentially, and stored in adipose tissue if not needed for ATP, meaning lipids, if we don't convert them into ATP, are stored in our fat tissues. Okay, so that's just the excess. Cool. So how do we do this? Well, we're going to catabolize them. We're going to call it lipolysis. Okay? Where a triglyceride is split into glycerol and fatty acids. So remember what a triglyceride is. Glycerol backbone, fatty acids attached to it. And it has to be done for liver, muscle, and adipose tissue. Okay? And it's enhanced by epinephrine and norepinephrine. For us to make lipids, okay, liver cells and adipose cells can synthesize lipids from glucose and or amino acids. This is what happens to all the extra food that you eat, right? It doesn't make you a bad person or anything like that, but when you consume more than your body actually needs, your brain goes, I might not have more in the future, so I'm going to take all of that and I'm going to convert it into something I can use. So you could eat 50 pounds of fish a day. Great. Think you're going to turn into Dwayne Johnson. Not likely. Okay. And what's going to happen is those amino acids from the fish are converted into adipocytes because you didn't get off the couch. All you did was eat fish all day long. Okay. So how does that happen? Again, it's the key, literally the key here is the most important part of this cartoon. This is the cartoon we just saw where you can take materials from the glycolytic pathway to make glucose, but also you can make other products here. And what I focused on inside class is the certain amino acids with insulin can be converted into acetyl coenzyme A to be entered into the Krebs cycle. Great, we can make ATP from it there. The part that usually freaks people out is you take triglycerides, go into the kitchen, go grab some bacon fat. Don't do that, okay? And you can break those triglycerides into fatty acids and glycerol. And here's what you want to pay attention to. Notice that you have green here for lipolysis. That lipolysis is stimulated by epinephrine, norepinephrine, and cortisol. So the green in epinephrine, norepinephrine, and cortisol, meaning stress-type environments, triglycerides are converted here and here. And then beta oxidation is what's going to happen to those fatty acids so that we actually make acetyl coenzyme A. It's glycerol that just is shunted now into this pathway so we can actually make glucose if needed. So the take-home message here is you can then take all of this. If you have plenty of what you need, your body can break it down inside the liver. You're going to make beta-hydroxybutyric acid here, acetone, and acetoacetic acid. These are the ketone bodies that people are talking about when you're living in essentially a ketosis state. For our class, definitely for our class, you want to make sure you understand the hormone regulation of the introduction into this pathway. So for us to make lipids, we need insulin. For us to break them down, we need epinephrine, norepinephrine, and cortisol. So where do those amino acids come from? Well, amino acids are either oxidized to produce ATP or to make synthesized to make new proteins. Remember, we need amino acids and dehydration synthesis to make all the proteins for our cells to function. Excess dietary amino acids are not excreted but converted into glucose. And that's why we call it gluconeogenesis or triglycerides, lipogenesis. Protein catabolism is when we actually break all of this thing down, but the important thing that I know I didn't stress in class, but I'll definitely stress during the review, is deamination is what actually has to happen. You got to remove that amino group, and for the purposes of chapter 26, when we're making concentrated urine, that amino group is converted into urea, and that urea is what we use to concentrate the filtrate that will be secreted into the bladder and ultimately removed from the body. Okay, so we actually put the amino groups to use because obviously ammonia is bad for you. Obviously, ammonia is bad for you. Okay. So what's going to happen? Well, I'm going to look at this thing right here, and I'm going to say, look, there are various points where we can add in amino acids for oxidation. Don't try to memorize this. I don't want you to. I just want you to see that the different amino acids that we didn't have you learn inside AMP1 now show up today. And you're like, oh, it would have been nice if we had learned those. It would have been. But no, more importantly, is you can see that Mother Nature's figured out a way that when you are actually starving and your body's starting to break itself down, it can still make ATP till you can find the next meal. The reason for this is this. Protein anabolism is carried out at the ribosomes in every single cell inside your body, right? This is actually how we make proteins. Remember? Bring the tRNA, binds the codon using its anticodon, dehydration synthesis occurs. Big deal. Okay? However, there are 10 essential amino acids in the human that must be present in the diet because they cannot be synthesized. That's why it's more important to eat a complete diet. Okay? And they put here beef, fish, poultry, eggs. I'm sure if you did some work, you could figure out other ways to get everything that you need. I am not knowledgeable enough to tell you exactly what the best meal is to get all of the amino acids you need for you, but definitely a healthy diet that has leafy green vegetables, even though it says it's an incomplete protein, is needed for other reasons. There are 10 non-essential amino acids that can be synthesized by what we call transamination. Literally think of it as Lego blocks. You take one amino acid, you convert it into another amino acid. This is a great place to pause, because what I want to do is I want to sort of finish up chapter 25. So it's going to require 4 sets of slides for us to go through, but that's okay, because it's been a lot of information on one chapter. But ultimately what we're doing is, at the end of the day, some of these molecules have other responsibilities inside your body, and I want to go over how Mother Nature actually regulates that. How does she decide when you need ATP versus you need to make a new protein? Okay? And your textbook doesn't really answer that for you. But essentially you have to remember the hypothalamus needs to make those critical decisions. Do you have enough energy to stay alive long enough for you to actually get, say, more energy in the future? Okay? So we'll pause here. Process this. We still seem to be having problems with closed captioning. I haven't yet been able to figure that out, but as soon as I do, I'll make all the adjustments.